All right, let's get started. Welcome, everyone, to the fourth and final presentation of McLaughlin Gardens Winter Lecture Series. We've had quite the journey this season. We started by learning all about trees and how they help us in our urban environments, and also what we can do for them to keep them healthy and happy in those environments. Then we moved on to gardening for ecology, so utilizing native plants to support our environment and boost biodiversity and provide for wildlife and pollinators. Then just last week, we learned how to identify all 9 of Maine's native frog species and where we can find them. So we are finishing off the series by taking a foray into the world of ferns. We have Sean Jalbert here today to speak to us about the secret lives of native ferns. But before I hand things over to Sean, you all know the spiel. We can't do this kind of low cost and free programming without your support. So if you would like to make a donation to the McLaughlin Garden, it would be much appreciated, and your donation would go a long ways towards supporting our programming. If you're here in person, you can make a donation by using the donation box in the kitchen, or if you are tuning in through Zoom, you can donate through our website, the big donate button in the right-hand corner of the site. So thank you very much, everyone, and hand it over to you, Sean.
Days after the presentation is complete until I get it uploaded on there. And it doesn't have quite the dynamics that the live presentation does, of course, but at least it has all the content there, the pictures, the descriptions that go on there. So it works out to be a good reference tool. A little bit more about my operation. I live in Elford, which is probably about an hour and a half or so south of here, down in the central portion of York County, just the very southern tip of Maine. And I started off as a native plant nursery. My main goal was to be sure that folks who are interested in native plants had a place to get them. In the early days, about 25 years ago, there was beginning to be some promotion of native plants, but people were running into roadblocks like, okay, you've got me sold on native plants. Now, where do I get them? Like, that's a good point. So anyway, so that's how I got into it. Then, of course, over the years, as businesses transform and you've got to kind of go where the demand in the marketplace is. And as Corey had mentioned as well, a lot of that now is consulting, going to people's yards. And oftentimes it's almost like we just want to take a walk through their garden, and maybe they're concerned about invasives and how to, which plants are invasives, which ones do I need to remove, how do I remove them, how do I get more native plants in my garden, et cetera, et cetera. So most of my plants are grown from a combination of various growers in the region, and I starting to grow instead of kind of growing everything that everybody else is growing, I'm focusing more now on plants that perhaps not everybody is growing kind of as a specialty. So this was the course description. You might recognize it there. Basically kind of reiterating what we already talked about, the fact that we're all familiar with ferns, but I since I was a child, I've always scratched my head. It's like, okay, so what are you doing there? I mean, we can see the flowering plants. They have a flower. We have pollinators coming to visit them. There's herbivory on the plants. We can see their interactions between the rest of the native plant community, but ferns are just kind of there. I mean, they're beautiful and we stop to admire them, but what's their ecological role? So this has been kind of an on and off lifelong study for me. And every year I learn something new, either in the book or on classes, and then I kind of go out to try to apply that. So interestingly enough, they have quite a influence with microclimates, and we're talking like on the square foot level, like underneath the leaves. I read an article the other day saying that ferns can bring the surface temperature down up to 2 degrees, which doesn't seem like much, if it's 80 degrees out, it's 78, but to the organisms that are depending on that cover or in those particular environments, that means a lot. It's certainly better than being 80 or 82 out, which more heat is kind of an opposite problem. So this is a term that's perplexed me for literally my whole life, that I've always been interested in plants. So this alternation of generations concept, which probably most of us have heard about, maybe even way back when we were in school or through our advanced studies like we're doing here, but always had a hard time figuring out what exactly do they mean. It just never stuck into my head. But I had this really nice book on fern ecology, and there was a section in there, and just the simple change, the alternation of two generations. That makes a lot more sense to me. Just the addition of that word too. So this is purposely kind of grainy and vague, and I didn't want to overwhelm with all the details from the particular life cycle here. But really, when we refer to this alternation of two generations, we're talking about the adult phase, which is known as the sporophyte. So that's the phase or the part of the alternation or the generation, if you will. But when we're walking out in the field and we see a really nice ostrich fern, or maybe we have a lady fern growing in our yard, or a shady nook with a maidenhair fern. That's the adult stage. That's the sporophyte stage. The stage that we never would see unless we were really looking for it quite hard is this so-called gametophyte stage, the other generation, generation number 2, if you will, which is much more difficult to find, even when you have adult ferns around. So that's something that's always perplexed me, and something that I've really devoted a lot of time to study and observation. We can learn and read books and come to classes like this, but for me, it's experiential learning. We take this, we go out in the field and say, okay, based on what we learned today, let's try to find fern gametophytes. I mean, what else do plant geeks do, right? We go out to try to find these things. So, and it's taken me years to actually figure this out. So I thought this was kind of interesting. I Googled alternation of generation for ferns, and and of course, everything on Google, there's endless screens and endless iterations of, all kind of showing the same thing, with the alternation of generations, the adult, the sporophyte, and the immature stage of the gametophyte. It's just, some of them are just downright impossible to figure out, like that one. But some of these you can kind of get the gist of it. And even though there's this great diversity of diagrams and ways of trying to portray, I mean, it's obviously a concept that's not terribly easy to get, or there wouldn't be so many ways to try to interpret it, right? That's kind of one way of thinking about it, at least in my mind. So I chose this one here. I like the simplicity of it, and it really just kind of gets to the meat and bones of the issue, if you will. So I added to it a little bit, and this is kind of blocked off here. But so essentially, the parts that we're probably most interested in, that we have the best likelihood of seeing, are in the red ovals here. The so-called gametophyte stage, which is considered to be the immature stage, and then the sporophyte generation, which is, again, the adult generation that we come across out in the field. But this is the other part that probably most of us have seen here, is on the underside of most ferns will, at a certain time of year, we'll see these little brown dots forming on them. And those are the structures where the spores are forming. And over time, those spores will get released, sometimes in the millions, sometimes just in the thousands, but just this huge, almost like an overkill of reproductive material going out there. You would think the heck that we would be overrun by ferns. We certainly aren't. I mean, some areas have a prolific number of ferns. Like if we're in like a moist area that has some shade, we'll see a lot of ferns in there. But for the sheer vastness of spores that are produced, you think every square inch of our landscape would be covered with them. And of course they aren't. So that just tells us nature's a harsh place to get going, especially for immature organisms. So there's very few that actually make it around and come down to the gametophyte stage. And then even, we'll talk about this a little bit more, I've been trying to culture ferns at home in the nursery, that it's very easy to get to this gametophyte stage. You can almost think of it as like planting a seed. A spore is sort of like a seed in terms that it's a dispersal agent, a dispersal mechanism. It's very easy to get to this point, but come to find out, it's not so easy to get to that point. We can have thousands and thousands of gametophytes, and sometimes only have one or two that will actually make it to the sporophyte stage. So this is something that I noticed very early on. I was, of course, ecstatic with the fact that I had my spores turn into such a green culture, but really kind of disappointed. What am I doing wrong? Why can't I get these to go, form this little sporophyte that's popping out here with this leaf and get to the adult stage? And evidently, it's not just like a cultural problem that I'm experiencing in the nursery. It's actually what we see in the wild as well, which would kind of, using that concept, explain why we're not overrun by ferns. The fact that it really takes thousands, tens of thousands, millions, maybe just to get one adult plant, which in turn can move to adulthood and get into the point where it's making spores. So, and we can get into all kinds of different concepts here. If you remember back to the basic biology with cell division, with mitosis and meiosis, Mitosis is simply just cellular division with a full complement of chromosomes coming along with it. Whereas in meiosis, that's usually relegated to the gametes or the sexual cells, where we're having a division of chromosomes. Half goes to it, as opposed to just basically duplicating or copying like we see in mitosis. So I didn't want to get too much into that quagmire. Of course, as in biology and any specialized field, in botany in particular, we have an incredible vocabulary that will drive you crazy. That's why we have botanical glossaries and dictionaries, because it's such a specialized verbiage there. So this was a last minute addition to the show, the slideshow that I put in this morning. It's like, okay, where did this alternation of generations term come from? So I typed that into my Microsoft Copilot AI generated wizardry, and it popped this out, and I did confirm it through various other sources. But the concept of alternation of generations was introduced a long time ago, back in the 1850s, by a German botanist. And he was really the first one to so-called revolutionize botany by demonstrating that all land plants share a life cycle of these alternating haploid and diploid phases. And even the flowering plants do it as well. They just do it in kind of a different way. And, this kind of goes on and on here about his life's work, and it also applies as well to those lesser known plants, certainly not less important, maybe arguably less important to humanity, but certainly not to the natural world. But ferns share a lot of the same reproductive features and characters with bryophytes, which are the liverworts, the hornworts, and the mosses, and some of the seed plants as well, although like in a different iteration. So these are two species of fern that we've probably seen before in our travels in the woods and such. One of them is interrupted fern, very common, fairly tall growing fern, can get to a couple feet tall, growing on shoulders of roads and wet areas, and really a pretty common species. And the other one here is cinnamon fern, which is maybe a little bit more selective, but again, we find it growing in some of these open, wet areas. Both, fairly tall ferns that can get 2 to 3 feet tall, and very obvious to us when we're walking through the woods. So these are interesting in a way. They're actually, one of the old-timey names for them was the flowering ferns. I mean, we know that no fern flowers, but if we use our imagination, maybe those look sort of flowery, maybe. But those are actually the fertile fronds, so-called fertile fronds. So they're specialized fronds, slightly different from these, and they have the spore-producing organs in completely separate fronds, as opposed to the other, perhaps more common method, which we'll see in a little bit. But this kind of gives you a good example here. Cinnamon fern gets its name because once these fertile fronds start releasing their spores, they turn a beautiful cinnamony color, kind of that nice, rich, warm brown color. And interrupted fern, of course, gets its name because you start off with these, the typical leaves, and then all of a sudden you have an interrupted portion where those specialized fertile fronds are producing their spores. So, those two names are pretty commonplace. I'm often scrutinizing names, like how the heck did that ever come up with the common name for it? And there's an interesting story to all of them, if you can find it and if you dig in far enough. So this is a much more common situation here where, again, going back to our specialized vocabulary, I mean, we can refer to this as simply as a leaflet on the stem, but if we use our vocabulary, refer to this as a pinnule. This one little leaflet is a pinnule, and we have many pinnae on the stem of the fern. So these brown spots that are on the back side here, we do have to kind of flip there's a adaxial surface and abaxial surface, again, getting into that specialized vocabulary, where we usually find these on the underside. So these are spore-producing cases, and these are probably a little gone by just because they are brownish and almost crispy looking. So that indicates to us that there's a very good possibility that those spores are no more. But typically, they produce so many spores that if we found this and we wanted to try to get some spores out of it to maybe try to grow ourselves, we could find some. So before we get too deep into this, just trying to go over some of these more eluding concepts sometimes because we can't even see them. But if we use a dissecting scope or even a hand lens, we can see a lot of these features. So these usually circular, elongated dots that we find on the bottom side of the fern leaflets, those are referred to as a sori, plural, sora, singular. And if we, picture this at like 10x magnification, if we go to this, say it's like 40x magnification, we can start seeing some more intricate details. The so-called indusium, which is like a very thin, probably like a one-cell layer sheath that will often cover that whole sorus for protection until the spores are ready to mature and ready to disperse. So can't really see them very well here, but there are these coil-like structures. Those are known as the sporangium. And within the sporangium, we have spore production. So there's very different layers that go in there and a very intricate system that's been honed over literally hundreds of millions of years. It must work, right? Because nature doesn't keep anything around that doesn't work very well or isn't effective. So a non-artistically drawn picture here, this is just a photograph, but if we're to go out in the field, this is what we would see on the underside, again, of the fern leaf. And, we see all these little dots. And again, these are kind of round, so we know that most of the spores have been released. What we want to try to do is get them while that sheath or that covering that we were just looking at, that indusium, is still kind of clear and covering that. So we know at that point that the spores haven't been released. And it's kind of interesting sometimes if we're looking at these under the microscope, we have inadvertent heat source from the light of the microscope, and these things will actually start to uncoil and release spores as you're watching them, just because of the heat from the desiccating nature of the heat from the lamp of the microscope. So that really kind of surprised me. I thought there was oftentimes when we're looking at things through dissecting scopes, there's other critters that come along with the plants, and that's what when I started seeing that, I thought it was, but come to find out, in fact, that it's actually these coils releasing and spores coming out. It's not like it's blowing back in your face or anything dramatic like that, but that's how these things happen in nature. Oftentimes, as structures dry out, they either open up or grow. It's kind of like a milkweed pod, that once those dry out, poof, they kind of open up along the suture and release their seeds. So this is at 10x magnification, again, through a dissecting scope. My son showed me how to do this. I never in a million years would have thought this would work. So I don't have a camera on my dissecting scope, but with a scope, if you take your cell phone, and you get it just right, you can actually take pretty darn good pictures of what you're looking at, which, wow, so I use it quite often. So when I was making this presentation, I was digging back through here in the archives, and I found some really cool pictures. So at the 10x magnification, we're starting to see some detail here, where the sorus is here, the various sori. You can see a little bit of the indusium here. That's kind of that brownish thin sheet that was covering them. Zoom in some more to 40x magnification, and we can really start to see these coil-like organs known as the sporangium. And in those sporangium, that's where the spores are being produced by specialized organs in there, where they'll be released. So you can even see somewhere this indusium here. And I added this up here just to give us an idea, again, just to kind of refresh on that vocabulary. I happen to be a very visual-oriented learner, so the more pictures within reason that are for easy reference, the easier it is for my brain to kind of make sense of these things. Interestingly enough, and we can kind of see why it would be here, and we'll see some diversity of it, every species of fern has a different arrangement of spores. Everything from kind of like these unique designs that we see here that are almost like leaf-like in themselves, to the arrangement of them. And then if we were able to look at it more closely look at just a hand lens or even just a magnifying glass, you don't need much. We have these beautiful arrangements of spore structures. And that's one of the key ways that we can use to identify ferns and to identify it to the species level. This one here is on Christmas fern, which is one of our evergreen ferns. And these are spore that have already been released. And there's either some mite or some spider web in here. It's invisible, but there was probably still some spores that are hanging out in there. But these will produce abundant spores, we'll say. Hard to know in terms of the thousands that it makes, but it's a very good fern to work with to get spores from. You'll see a lot of rock polyploidy in my presentation. It's just a fern that I admire for its tenacity, and it's also called the resurrection fern because it grows typically on these ledgy, rocky areas or these glacial erratics where When we're having a drought, it will be shriveled up and dried up to the point where it certainly has to be dead. And then we'll get a good rain event, and they're coming back out just as happy as can be, green and verdant. So they certainly earn that other name of resurrection fern. But these spore cases are really almost multi-dimensional, even just to the naked eye here. I mean, you can kind of see them almost popping a little bit along the pinnae. So that's an interesting one, again, to study and to really see. So we're assuming at this point we've seen what these spore-bearing structures are like, and the spores have been released. Okay, what happens next? You're certainly not going to be able to find spores out in the wild just because of the fact that they're, borderline microscopic, and they kind of disappear into the leaf litter so quickly. So I've gone out looking for these, and it makes perfect sense. The best place to look for these young fern stages is where the parents are, right? Where there's suitable conditions. So this particular picture here is really interesting. And this, it's hard to kind of get the scale of it. But I mean, this is an area that's probably about that big. It's along the embankment of a stream, so there's consistent moisture there. But we see most of the fern life cycle here. I mean, we know that there are spores here, but this so-called gametophyte stage, which is like the immature stage that we're discussing, that's there. At that point, the chromosome number is just half. At this point here, we see the sporophyte stage. So we're seeing a kind of like still an immature fern, but it's kind of made that leap into adulthood. And these are probably maybe about a year old, and they have that double complement of chromosomes there. And the more we look, you see some more this leafy tissue that's formed at the gametophyte stage. It's referred to as thallus. It's usually a few cell layers thick, and it's kind of green, and it looks almost lettuce-like. And I'm not sure if you could eat it or not. We're talking about eating certain ferns here before we started the presentation, but they almost look good enough to eat. So I had already mentioned this before, but something that I ran into in culture and trying to get these spores to grow is just this very frustrating, disproportionate ratio of emerging sporophytes, these guys here, versus just the sheer mass of the gametophyte stage and the thallus. And it took me a long time to put two and two together. The fact that I thought, again, like I was saying, it was something that wasn't quite right in culture. I mean, we like to think the nursery setting is an ideal world, and it is in many respects because you get plenty of light and food, water, and all the good stuff. But sometimes it's by taking it out of context and out of nature, we're missing factors that we don't even know enough to know that we're missing, which I thought was kind of happening, which probably is still the case, but it's happening in nature too. Where we have, I don't know what the numbers are, probably hundreds and hundreds of the immature gametophyte stage, and really relatively few adult plants that are actually making the jump into adulthood. Of course, usually when we find plants growing out in nature and communities, we find an awful lot that's going on. So in this case, we have liverworts that are growing with mosses. We also know that we must have had some spores to get in here, because these are our immature sporophyte, basically adolescents, we can think of them, and hopefully someday they'll turn into adult ferns to be able to continue the cycle. So again, in this particular situation, this is a kind of a piece of rotting wood, and it's probably not much bigger than about that. But at the risk of beating this concept to death, I was just so elated to be able to finally see this going on in nature that I try to take good examples, pictures of good examples when I can. So again, we see kind of a little bit of all three stages going on here: the young gametophyte stage, and the this is probably, it goes like 1, 2 with the sporophytes, and then we see some plants that are kind of ideally going into adulthood. So that's an interesting picture there. So something that I always wondered about is like, okay, how can you tell the difference between fern gametophytes and liverworts? And if we want, again, to get into the details, there's a lot of rather subtle differences between them. But in this particular situation, this was really easy because ferns don't produce reproductive structures like that. liverworts do, and the hornworts as well, and mosses are kind of similar in that vein. What's really interesting about this is that ferns spend most of their lives with that double chromosome number in the adult stage. Mosses and liverworts and hornworts spend the vast majority of their lives in what we consider the immature stage and the gametophyte stage. So with liverworts, this whole structure here, which is the structure that's the longest lived and the one that we're most likely come across in nature, is the haploid phase. It's got that half number of chromosomes, and the only part of this whole plant that's diploid, it has that double part of chromosomes, are these structures here. So it's just the opposite of most other organisms. So how did that ever evolve, and why did it evolve? And as I said before, obviously it must be working. I mean, these species and plants are still with us after all these years. So again, we spoke of mosses in terms of having the relationship to ferns, and that they have that alternation of generation cycle as well. And again, just like we were talking with the liverworts, most of what we see here on the moss has got that single chromosome complement, and is what we would almost consider to be the immature phase. And these structures here, that's where the spores are, and that's where the so-called diploid phase is. That's the only part of the plant that would be considered the sporophyte. We already talked about liverworts, of course, ferns, but the so-called princess pines and lycopods that we've all probably seen, talking about mysterious creatures of the woods. We've all seen them, and we have a fairly large number of species here in the state, occupying a variety of habitats, even some habitats that are in fairly early succession, which is kind of interesting. You would think that we'd see a lot of these plants in just older, mature forest, where they have had 100 years, we'll say, to set up after the last disturbance, coming from, like, say, ashland back to forest. But what's really interesting about the lycopods and the princess pines here is that they have subterranean gametophytes that don't photosynthesize. So studying those has been tremendously difficult. And we've only been able to really kind of starting to get a good idea as to what's going on there with the advent and the efficiency involved with molecular sequencing, and able to get biological material. We don't know what it is, but we can rapidly sequence that out using fairly widespread lab techniques now. Compare that to a data bank of DNA sequences of those known organisms, so we can see what's going on in the ground. There's been some really interesting studies of that. Even not only studying the plants themselves, but the mycorrhizal fungus associations that we find, especially with these here with the underground gametophytes. I've been interested in trying to get those going for a number of years now. Apparently, it takes anywhere from 5 to 20 years for the gametophyte stage to get to the sporophyte stage, which is that part there. Which who's got that kind of time? Again, just before we leave the topic here, kind of a side-by-side comparison so we can see liverwort on the left and fern gametophyte on the right. So now that we have them side by side, they don't look as similar as we thought they did when we didn't have them on a side-by-side comparison. So this is a really good shot here. This is just forget now how big it was, but a fairly large patch of fern gametophytes, and there was not a single sporophyte there. I mean, did I just miss it? Is it going to be coming in next year? Who knows? I don't remember even where I took that picture. I believe it's probably in the Gorham area, but that's just a kind of the last splendid example of the fact that we have, all these immature ferns, and they may never form mature sporophytes. And after a while, these gametophytes will die off and create room for other plants to come in and basically try their hand. This is a really interesting shot here where this is in a riverbed in between two cobbly boulders, and we see this sporophyte coming up here. And again, there's a few of the immature ferns and only one that's needed out of all of those. So eventually the gametophyte that gave rise to this adult plant here, that will die. and just kind of let the sporophyte go on by itself. So that's an interesting shot there. Fun botany fact. So I'm thinking, okay, so I read about these subterranean gametophytes and tropical ferns. Like, do we have this happening right here in our state? And we do, in fact. We have the fern family, the Ophioglossacea, say that 10 times fast, which is the family for the great ferns and that are very mysterious in the fact that they don't come up every year. You know, we could find them one year and go back 5 years every year afterwards and never see them again, and then they'll come up on the seventh year. So they're very tricky to monitor that way. We don't have a good handle on what we have in the state. You know, we know what species are here, but in terms of the exact population. So this other species here, the northern adder tongues fern, this sort of looks like a snake tongue. That has these so-called subterranean gametophytes as well. And as the white color would indicate, there's no chlorophyll there. There's no self food production. It's not able to do it. It uses mycorrhizal fungus association. So again, we can see the importance of these plant communities and needing all these components there to get from this stage to that stage or to the adult form where the cycle will just start anew again. So that's a pretty interesting fact. So now, do we have any questions at this point? We don't want to get too far into the weeds here.
I have a question, like, way back at the beginning, to make quick reference. How long does it take to get?
Well, of course, it differs for every species, but I would say, like, when we get our spore on the appropriate medium, anywhere from 2 to 3 months, we'll get the thallus stage, which is that leafy stage. And then it will take probably another 2 to 5 years to get to what we would recognize as a mature adult fern. So I have a hard time with getting to I can get from the spore to the gametophyte stage where we see that leafy green thallus. And every once in a while, I get really lucky and get to the sporophyte stage where we'll start forming the adult plant. I have a terrible time transitioning from that sporophyte stage to like a regular, like a pot size. And I always lose it at that stage. I've lost dozens and dozens.
When you were talking about the underground,
Yes,
it's like lady slippers, where you need those microorganisms in place.
exactly.
So
Yeah.
like with lady slippers, they tend to be like soft.
Right.
So they would, so your ferns would be similar.
Yeah, kind of the same idea because with orchid seeds, they have very little what's called endosperm or that starchy material. It's going to be that initial energy source or carbon source for that embryo as it's growing. So they really have to be around the mycorrhizal fungus, which basically acts as like a nursery of sorts and bringing them the carbon that they need. And I don't know how much of a relationship there is there, if that orchid seedling is giving anything back to the fungus. It doesn't seem like it, does it, at all. So I guess it's almost parasitic.
Some of them, there's some studies that show like after maybe like 7 years, some of these like micro parasites start to give something back to the mycorrhizae, but it's pretty one-sided.
Yeah, we do see that from time to time in nature. So
would you need to add that to your medium that you plant in the ferns and so that?
Yeah, probably without getting too detailed into it would be the best way to try to do it anyway is, if you have access to the habitat that the fern was growing in, simply taking some of that soil, and it doesn't have to be much, just a teaspoonful, and hoping and assuming that probably, okay, if that fern is growing here, then the mycorrhizal component must be, or it wouldn't be able to be growing here. So hopefully there might be enough mycelium or maybe some fungal spores in there that will kind of jumpstart the process. So I think that would probably be the easiest way of doing it. But it's definitely a fun thing to try, and I really encourage folks to do it. It's not that hard, and there's a ton of online resources involved with going through details, and we'll start going through these a little bit here, but on how to do it, and it's really kind of an enriching experience to know that you can work with the organisms that way. So collecting fern spores is very similar to taking spores or making spore prints with mushrooms. They both utilize spores for reproduction. And in this case with mushrooms, we'll just take the cap, and you can't see it very well here, but we have the gills with the mushroom, and inside those gills are the spore producing organs. And if we set the mushroom cap here overnight or for 2 or 3 days, we have these wonderful spore prints that we can use. And usually that's how I get the spores for my ferns as well. Not so much simply leaving it on top of the paper. You just walk by and you can lose all your spores because they're literally lighter than air. Typically what I do is take my fern frond and I'll put it in a just a paper envelope and just seal it up lightly. So as they start to dry out, those spores are going to be released and you can catch them in the envelope. And you have to we'll call it sporulate very, very lightly. It's almost like you can't do it lightly enough because there's so many spores coming in. Just a tiny little pinprick of spores has probably thousands and thousands of them, and you get a really high germination rate, unlike some of these flowering plants that we might try to grow that have a very low germination rate. So a quick section here on fern growing FAQs, or frequently asked questions, and stuff that I've thought about that I thought would be helpful here. So, when do you collect the spores? And like seeds, spores ripen at different times of the fern's life cycle in different parts of the year. Some of the ferns that we looked at earlier, like interrupted cinnamon and royal fern, those mature incrementally starting in about late May, going through maybe the first or second week of June, depending on what kind of a rainy season we're having at that point. So, which leads us into the next point of, how do you know when the spores are ripe? And it's kind of like the Goldilocks clause. You don't want to get them so they're too ripe because there's nothing there, or you don't want to get them when they're immature because they're not probably going to turn into anything. But usually they're looking like seeds and fruit. We're looking for some sort of color change, some sort of indicator to tell that there's something going on here. So the spores come in a variety of colors, from green to orange to brown to cream. So they're notable in that way too. So, typically if you see, we talked about those organs, the sori and the indusia. If those are brown, usually at that point you've kind of missed the bus, but there's always a few spores in there just because there's so many spores made. We often talk about with seeds, especially our native plant seeds, that we have to give them some sort of treatment for the seeds to break dormancy. So cold treatment or stratification is one of the more common things. But with spores, we don't have to do that. We collect them and they're ready to go right then and there. So that's kind of nice. It's sort of a little bit more relaxing because we don't have to go through these, what seems like this endless variety of dormancy breaking requirements. And it's really quite easy with those. So, and then the last part, well, how do you get the spores to germinate? So like seeds, they need access to light, moisture, and kind of a stable humidity levels and a suitable substrate to grow on. That's the part where there's some hard lessons learned when I first started, where you want to make sure that you have enough depth in your container to have that kind of capillary or wicking action when they're watered. You need that water to leave. If not, it just stays saturated and it turns into all kinds of messes, which you'll see in a few slides here. But so you want to have something that has adequate drainage. Almost every recipe, if you will, or methodology for trying to grow ferns from spores that I've seen, they almost always say to sterilize the medium, which I did dutifully when I first started out. I love to experiment, so I'm thinking, okay, what happens if I don't sterilize? Because we have this beautiful compost-rich mix that has very delicate organisms that are in it that are suitable and welcome for growing plants, and nutrients that are also kind of a complex macro and micro molecules that heat can probably break them down and they won't be as effective afterwards. So I started growing without doing any sort of sterilization. I actually had better luck. And to be honest, I was using a different medium as well, and different kind of combinations with some coarse material or drainage on the bottom to make sure that I didn't have standing water in a pool like I did when I initially started. Saw these ferns and slides earlier on, but the cinnamon fern, we're going to zoom in a little bit more, and we can see kind of that brownish orange color forming on it. So this is pretty much a perfect time to collect those spores because the spores are green in this case. So we're starting to see a lot of the green still there, but we know where most of our spores are mature, they're releasing. So what we do at this point is either just take a couple of these little subsections here, or just take maybe one stem, and there's enough spores to repopulate half of your town just in that part there. So with the royal fern, we can see these very distinctive fertile fronds, and those are still kind of like this bright verdant green, so they're not quite ripe enough yet. With royal fern, we actually want to wait until those fertile fronds are starting to almost turn like a blackish color, and then that indicates that we're ready to collect the spores at that point. So this looks big, of course, in the slide, but it's only kind of a tiny little section here. But these are some fern spores that are germinated in culture. And as you can see, I mean, there's just a lot going on there. And this is at the stage where I didn't have my drainage worked out and my types of media selection worked out as well. And we have quite a bit of sporophyte production arising from those gametophytes there. I wasn't able to get any of those sporophytes to get to adulthood. So this is my latest iteration. Instead of something getting more complex and more sophisticated, I'm almost devolving in a way of trying to make it as simplistic as possible with the things that we all have around the house. We all have a ton of pots. We probably have clothespins, and we have some sort of plastic wrap. And that makes a really fantastic containerized system to germinate our spores in. This is like a 2.5 or 3 gallon pot, so there's some depth to it. On the bottom part of the pot, if I remember correctly, like in this case, I had some coarse draining mix. Maybe there was some coarse bark in there, but on the very top of it, I was using just a readily available compost-based mix. I think it was Fox Farm mix. It seems to be kind of ubiquitous now in hardware stores. And as you can see it through here, I had some really nice sporophytes coming off. They were probably 2 or 3 inches tall. And sadly, they're all dead now because I can't get them up to the next stage. But I'll get there eventually, and it's all a learning stage. This is a couple of the more spectacular falls. And, let's face it, we probably learn more from failing than we do succeeding. I think because failing makes us go back and look at everything. What did I use for a container? Did sterilizing help or maybe hinder the potting mix that I use? Too much water, not enough water. Did I fertilize when I really didn't need to? So this is basically just a big shagadelic mossy mess at this point, and that was sterilized. And then in this situation here, I was really dealing with drainage issues. And at this point, it was just a number of different fungal species that just went bananas in there and took over the colony. And they started feeding on the ferns that were developing. And both of these had issues with fungus gnats. Of course, now you have fungus growing. The fungus gnats are going to be there, right? And they're terrible little critters to deal with because they're both in the adult stage and the larval stage. They're doing damage to your plants, whether it's actually eating the plants themselves or like in the larval stage when they're in the soil, they're feeding on the roots. And of course, you destroy your roots, your plants succumb quickly thereafter. So this last section here, we've got about 15 minutes or so left. We're going to be looking at fern ecology and how ferns anchor forest soils, influence microclimates, and support biodiversity in the understory, which we've already touched upon a little bit here and there. I have a tendency to get a little too far ahead of myself sometimes during my presentation. So this is my son hiding. I was trying to get some pictures of ferns. He's like, "Pop, look at me!" And, well, that's pretty cool. We can use that at some point. So of course I integrated in here. The dense fern growth for create habitat for creatures of all types.
And the fern is that tall? Like how tall is he?
Oh, so he's a little taller now. He's probably about chest height now. So he's actually crouching, kind of like that. So those are pretty tall. I mean, we have mostly interrupted and cinnamon ferns in there. So I mean, conceivably, some of them could have been over his head. And also, I mean, not only is he crouching, but I'm up a little bit. I'm kind of taking the picture from looking down, so it has even more of a dramatic effect. So. So this is a kind of the opposite picture. So this is a picture from the ground up. And I mean, this looks like a fern forest from back, in the Carboniferous period before the dinosaurs. But, again, we're probably about that tall, but you can kind of get a good idea as to everything that's going on under there, with the shading and the increased humidity. And I'm sure it's probably a countless number of organisms, both plant and animal and fungal, that are using this. And the ferns are kind of the, moderating those habitat features, and they're really kind of ruling the roost there. Probably most of us have seen these gigantic patches of sensitive fern, and we typically see them in areas that are open fields. Sometimes we see them creeping into the shade of the forest, but usually we find them in kind of disturbed areas. Well, let's face it, a field or a lawn, it's a disturbed area. It wants to be forest, and we're not letting it. So we see this prolific growth of sensitive ferns, and sensitive ferns have been shown to be able to take up a lot of excessive nutrients that are in the soil and hold them and basically retain them from being maybe flushed away or being used by other plants. So the more we start studying about ferns, we realize that even though we don't have, say, pollinators visiting them like we do the flowering plants, they're really controlling the chemistry of the soil to an extent that you couldn't have imagined unless studies were done to try to investigate these things. So this is kind of in the wet section of my field where I live in Alfred, and we have this massive layer of sensitive ferns, but we can see we have multiple species going on here. We have these royal ferns that are in there, and there's cinnamon and interrupted as well. So this is on the edge of a pond, and we often find royal ferns very elegantly just kind of hanging over the sides of the edges of these kind of mucky soils that could very well be destabilized if those plants are there, because they have some really, they don't have the root systems of like a tree or a shrub or even a flowering perennial, but they have these very thick mats that form, and that kind of holds everything in place. I'm sure they're utilizing some of maybe the excess nutrients that are available in the pond water and locking those down so they're not being flushed into the water column where they're causing algal blooms and other problems with clarity and just making the ponds kind of unfit for a lot of different other organisms. So kind of going from wetland shoreside to more of an upland situation, these are beach ferns, of which we have 2 species in Maine. These form really dense mats in forest settings, and spend quite a bit of time up around the Moosehead Lake region in Greenville. And it's very common to see these growing in large areas on hillsides and in amongst moss. And again, I'm sure they're influencing the soil dynamics there as well. There's been very few studies that have been done on those, especially it seems like a lot of the research done on ferns is done on tropical ferns. And we're finally realizing that, hey, wait a minute, we need to spend some more time studying our more temperate species because there's an awful lot going on there. So ostrich ferns, that's the source of fiddleheads. We find these growing in very dense stand clumps, but dense stands nonetheless in these floodplains and forests where they're really doing well in that kind of rich alluvium that's maybe comes every few years from flooding events. And again, kind of holding down the soil in this situation from causing even more erosion and losing more down the river. So this is in Rochester, New Hampshire, which is fairly close by to where I live. Interestingly enough, ostrich fern and sensitive fern are in the same family. You can kind of see that in the plumes where the spores are produced in the ostrich fern. That's how it gets its name. And then if we kind of think about it, I don't have any good pictures of it, but sensitive fern has somewhat of a similar spore producing mechanism there as well. So never would have thought we'd find ferns growing right along the ocean. It's kind of deceiving here. I mean, the shore is probably, I don't know, 30 or 40 feet away at this point, but this is on the marginal way in Ogunquit, where I've done a lot of ecological restoration work down there. And it's a very disturbed area. It has been for at least 150 years, probably more. And there's a lot of it. There's mansions on the top part of it here that are kind of lining the water edge, but there's still kind of a buffer there. And the hydrology has been really changed a lot. So we have a lot of these seeps and water drainage areas that probably weren't there before all these houses were built. But nature being the innovative concept that it is, now if we have some moisture, well, let's grow some marsh ferns there. And doing some research on this particular species, they're not uncommon to be found in saltwater estuaries and marshes. So it kind of makes sense that they're growing there. Bracken ferns are a force to be reckoned with. Supposedly growing on all 7 continents. Really, Antarctica? Who am I to judge? I've read that in a number of different places, but they do have a global distribution, and but a lot of areas they've actually been introduced in, and they've turned into invasive plants at that point. And the really hard thing about these is that they do have cancer-causing chemicals in them, and they have a really hard time with this over in Scotland and the greater Great Britain area, where these will be growing up, and all of that area has been disturbed, has been occupied by humans for thousands of years. So these ferns love disturbance. They'll be growing in pastures. The cows will come and eat them, and they'll develop bladder cancer. So that's a very strong correlation there. But these are very strong successional drivers too. Again, we're up around the Moosehead Lake region, but there was a whole gravel roadside that was just as thick as you can imagine with these things, and excluding a lot of other plants from establishing. Hay-scented fern sort of does a similar thing in the fact that, again, here along the roadside, and we have a feeble attempt at lawn. This is a gravel road here, but these hay-scented fern they're raring to go, and they can grow quick. They've adapted to these early successional disturbed sites. So if the spore comes in and grows into an adult plant, they can go off in a hurry. And their main method of spreading is by underground runners. So there's a lot of landscaping applications for these plants too. So we can have ferns in sunny, harsh spots in our landscape that are, we typically wouldn't associate with ferns. We think, oh, ferns need shade and consistent moisture and rich soil. Not these guys. They thrive in these conditions. So this is another, hay-scented fern rapid outgrowth. This one is in York, Maine, south of where I am. But there's been studies that have shown that hay-scented fern will keep species like red oak and black cherry from starting. There's no sort of allelopathic connection. It's not like it's secreting a chemical that's killing the seeds or preventing them that way. The simple fact that this is growing so thick that when the seeds of these trees happen to land there, a bird comes by and poops it out, or a squirrel drops it off, or what have you, it doesn't have access to that consistent moisture that it needs and that top layer of soil. So it's not so much, I guess you could say it is a plant, but it's the growth habit of the plant that's excluding these other species from starting. And because of that, it's affecting forest dynamics. And I've seen that in a lot of places, but didn't really put 2 and 2 together, where you'll have an area that there's been timber harvest in, and you go back a few years later, and usually it grows up very quickly from water sprouts from the stumps or other plants coming in. But you see this in like all the skidder paths and the areas that have been opened up, and you go back a few years later, and wow, there's still not much for tree growth, and find out there's actually this scientifically validated correlation that it does prevent some species from taking root. So kind of switching gears here a little bit, we'll try to finish up. So again, polypody fern, we often find it growing on these glacial erratics, and all it takes is one spore. It may land on a little patch of organic matter, some leaves that have accumulated in a shallow pocket. And as the years go on, how old is this patch? Is that 100 years, 200 years, 50 years? I don't really know, but and we can see the leaf litter being accumulated there. So all these kind of forces working in conjunction, it's actually generating more soil on that rock. So as time goes on, that rock will be kind of broken down and made into a suitable habitat for a bigger variety of plants. Growing right along these thin cracks that you can't even slip a piece of paper through. They're so small, but again, all it takes is one spore. Polypody fern and Christmas fern are at least two species of ferns that we have that are evergreen. This one I took just over the weekend, or we had some snow down there in Alfred, and I just thought it was kind of a good way to illustrate the fact that these ferns stay green throughout the season. So woodsy fern growing on hardly any material at all. And the blazing sun, at least polypody fern, grows in the shade and is able to kind of recoup some of its moisture losses that way. But this woodsy fern will grow in the blazing sun, and seems like it takes it. It's another shot of a nice big group of polypody fern literally growing on the side of a rock face. This one here we don't see very often, maidenhair spleenwort. This picture I took in the so-called Grand Canyon of Pennsylvania, which is in north central Pennsylvania. And we can kind of tell this shelving here, that's limestone, and it was dripping wet, and the moss was there. So it loves these comfy little pockets that it can settle in. And of course, likes the higher pH that limestone soils will give. I had a customer request to come out, and it was in Medford, Massachusetts, and she had a splendid backyard. I was jealous of when she had a double lot type situation, and she was very artistic and creative the way she had her plants arranged. And she had 3 or 4 different species of ferns that were containerized, and she overwintered in that way, and they came back every year. So I thought that was a really kind of a clever way to get more ferns into your garden. And if you don't like them there, you simply pick them up and move them to a different location. So that's pretty convenient too. We're going to be here pretty quick, right? Another 2 or 3 weeks. Or one of the characteristics of most ferns are these croziers or shepherd's hooks here that are tightly coiled, and these will emerge out of the ground and very elegantly unfurl. Sometimes if you have a warm day, they'll come out really quickly, but that's kind of the aesthetic example of the aesthetic beauty of ferns at all stages of their life cycle, whether it's very early spring or further on in the season. So that concludes the entertainment portion of the presentation. And as always, if you have any questions, you can contact me at email, or get on the website, or just even do the old-fashioned thing, give me a call, and we can talk plants. And certainly, if there are any questions or ideas that came up during the presentation today, I'd be happy to talk about those down. So, yeah.
Now you sell ferns at your.
I try to.
Because I was going to say, how do they do a transfer?
Once
they're established.
I think better than those intermediary stages that I was discussing, where we're going from spore to gametophyte and then up to that. That's like the really critical stage. But when they're containerized, they're a much more stable plant. And what we like about containerized plants is plug and play, right? You just put it in the ground as long as you have a reasonable place to put them.
So if you do buy the pots, if you do buy the potted plants, you can into your garden.
Right. Yeah, typically it's much more successful that way. So, something like maidenhair ferns always touch and go, but something like the wood ferns, for instance, or Christmas fern, those are pretty hardy and have a good degree of success associated with them. Yeah, and
prehistoric ferns, tree-like, go back to the dinosaur age.
Yeah, so there was a ton of carbon in the atmosphere back then, as the Earth was going through. We're at that, like, even before the dinosaur age, like in the Carboniferous period, that was like what, 300 to 400 million years ago. And Earth was kind of going through the geomorphological changes, and everything grew big back then, plant-wise, because there's so much carbon. There was basically unlimited resources to grow those plants. So from what I gather in the research that I've done, I think that arguably or probably the primary factor, there's just so much raw material to work with. So, and then as we kind of went in through the Carboniferous period, and these gigantic ferns that were seemingly stories tall, and who knows how big around, once that carbon was pulled out of the atmosphere and locked up into those, and those kind of died and settled out, and yielding to the next epoch or the next series of organisms that were going to be dominant. They got buried and eventually turned into oil.
Oil, exactly.
Yeah, so burning ferns. The Carboniferous. Yeah, burning ferns with the Carboniferous. Yeah, that.
Teenager, I was for fern. Ferns cover it with
plant to go down there, and that if you could do it that way, spore by spore, because like I was saying earlier, it's almost impossible not to get a zillion spores on your little container. So by doing them that way, perhaps he had better success because he was able to space them out almost again, like we're always comparing to seeds and such. But where we have a container and we just don't dump in 100 seeds, we place our seeds rather strategically. So there's like everything, there's a.
I don't know if he placed seeds, roots, or
Sure.
cup,
Yeah.
cups.
Yeah, from a kind of a different perspective, commercially speaking, ferns are of course being grown, but they're being clonally produced using plant tissue culture.
Yeah.
And there's just a few places in the country that have it scaled up to the point, and they're pretty much supplying most of the nursery stock material in the country from the investigative sleuthing work that I've done to try to find the plant source. And they can produce millions of each species quite easily, and they're probably unlimited by space in that situation. So ferns aren't grown by spores on a commercial level, just because, as we've seen here, it's not impossible, but it's really hard to get that scaled. And each of those really delicate stages that they're going through and trying to step them up into larger containers is very difficult doing it that way. So, but not to say, you could speak with somebody else and they could say, well, it's easy to do, so.
So I'm wondering, like, when you go to a greenhouse and buy, what method are they using?
Yeah, so of course, every situation is slightly different. I mean, hopefully they're not just yanking them out of the ground. Depending on the type of nursery it is, but chances are, as is the case with most plants, they've gone through about 3 or 4 different hands before they come to the final point, which is you in your garden. So there's a good possibility that they're being clonally propagated. It's highly unlikely, unless it's a small specialized grower, like that fellow there, that they would be grown from spores, because it takes an extraordinary amount of patience and luck and skill and fortitude. And it may take you 4 or 5 years to get 10 plants, and you could sell all those in one season and not have any more for another 4 or 5 years. So long story short, they're probably clonally propagated. Yeah, so it's not a bad thing. It is what it is. I mean, if we didn't have that sort of technology available for scaling, then we probably wouldn't have access to them at all. So we've got to kind of use what we have. And if we want to make the system better, we roll up our sleeves and we try to do it ourselves. Sure.
We have a few really large. It's one plant and it spreads out like that. And it's not this big. Just
if it's like in an upland area, I mean, it's probably something like a cinnamon fern or interrupted fern, or if you want to.
Even if it's growing that large.
Yeah, I mean, because those are probably our largest species of ferns. They'll grow sometimes chest high in some situations. So, if at some point, maybe if when they're back up and out again, if you want to take a picture of one and send it to me, I'd be happy to look at it for you. Yeah,
I have one that grows in a very dense, wet area, and then another one that looks just like it to me, but maybe not, is in a light area, but it's still damp.
Yeah.
In that area.
Yeah.
But to me,
yeah, I mean, they could very well be the same, or they may be just a slightly different species. Yeah. Yeah, hopefully, if certainly not meaning for you to memorize all these litany of infinite vocabulary words today, but just get a better appreciation of the complexity of what's going on there. That I didn't have any idea as to what it was, and I read about it in books, and that wasn't enough. I went out and found it and replicated it, to a certain degree. Just in addition, just to kind of see it yourself. So we don't have to be bystanders and what we learn about and what we read about. We can go actively participate in these things. I like to do as gardeners, whether we're growing vegetables or flowers or trees or whatever it is we choose to grow. So,
Some flowers that are like.
Right.
Awesome. So.
Yeah.
It's not the same.
yeah, 2 years they're done, and that seed doesn't sprout and they're really done. So,
And we don't have any questions on Zoom, but we do have lots of people expressing their appreciation for you speaking. Amazement of how many different types of ferns there are.
yeah, it's really an understudied field. And I think as humans, we seem to have this natural affinity towards the flowering plants because they feed us. That's probably the strongest reason why. It makes good sense, but there's so much more going on out there. And we're lucky enough where we live in Maine that we still have reasonably intact and functional ecology going on. So we may not have it in our yard, but we get in the car and go 5 minutes down the road and we can go to preserved land, or we can go out in the woods that are attached to a property next to us, or so on and so forth. So we're really, really fortunate that way that we live in a completely paved over landscape like so many other places. So,
Yeah, I was just thinking, I have a lot of Japanese fern.
Yeah.
And I painted fern, and I'm like, there's all these different types now coming out. And I'm wondering, they're not natural ones, right? But they do spread.
Yeah. Yeah, I mean, of course, we always have to be careful of naturalization. Like we plant something in our yard before we know it, it's here and there, everywhere, and it seems to be adapting to the situation, naturalizing. And of course, there's always a fear of, it takes that next step into being invasive. And I don't know of any invasive ferns that we have right now. There's a lot of ferns that are invasive in different parts of the world. Like Japanese climbing fern is a terrible invasive in the southeastern part of the United States, kind of like what kudzu and bittersweet do. It's just, going over these trees, forming these massive tent cities and just canvassing the place. And I was talking with bracken fern, that's straight up invasive in a lot of places. So, but as far as I know, I haven't come across any ferns that are invasive. So I think, in that situation, you'd probably be fine. Always good to keep an eye on it if they get too rambunctious to maybe herd them back, but it's probably be okay in that situation. So. Go right ahead.
We were living in Texas in the winter time, and one of their deepers had foxtail fern. Is that your fern?
Yeah, I'd have to look it up. I'm not familiar.
Really beautiful.
Yeah.
That
Is it green?
no, sorry, we Home Depot and it's called a foxtail fern, and
that this is all interesting, but there's so many different ones. And are they native? Are they, just brought in? And I guess that's kind of.
Yeah, so if you're out in the woods or in wetland or in a meadow looking at ferns, they would almost certainly be natives. So I don't think I've been in any areas that I know of that I've seen non-native ferns growing there. I can't say that it won't happen or it can't happen, but at this point, as we're speaking, it doesn't seem to be an issue. So with ferns, I mean, those are some of kind of like the easiest group because the pretty much everyone that you see is going to be a native species. And just start picking away at them. I mean, in all honesty, I'm not excellent at fern identification. I hate to admit that in front of you, but I've been working on it my whole life. And some of the groups like the Massachusetts fern, the New York fern, and some of the aspleniums, and it's like, it's just a matter of taking the time to do it. So going back to this foxtail fern, so yeah, it's not a fern at all. I mean, if it's it's in the genus Asparagus. So I mean, is it kind of look something like that? Maybe I'm not sure if that's a right. Yeah, foxtail
But they
fern.
were selling at like
Oh.
Lowe's and.
Yeah, yeah.
I told Jane when she got one, I said, that's not gonna live in me.
No, well,
You know, so
She brings it in.
we just keep it. It's in the basement.
yeah, we have a
It's a beautiful.
fellowship, yeah,
But foxtail fern is,
not a.
it's so great flowers, my throat.
Yeah, yeah. Yeah, there's a lot of plants like sweet fern as a shrub and it's not a fern at all. You know, it's a woody plant, so sometimes those names get thrown around, but they're careless. We had a question there.
Yeah, we just built a house and unfortunately, they had to remove a lot of ferns, a beautiful. I could see where they dug up when they excavated, where they dug up. I grabbed as many of them as I could and get them back in the soil, but but will some of them still come up even though that cluster is not there in the soil, or they probably only dug down like, I don't know, a foot maybe, when they excavated? Is there anything, how far down do the roots go where something might?
I mean, it depends, again, it depends on the type of fern that it is, and some of them like a hay scented fern or.
It
Yeah.
was interrupted.
Okay, so, yeah, I mean, those go down a little bit deeper, and so nature, thankfully for us, is very resilient. So I wouldn't be surprised to see something come back from that, especially where you kind of put them back there, that kind of black, wiry, fibrous rhizome that's there.
Yeah.
Those are pretty resilient because they're so thick. There's still nutrients and moisture in there, so chances are, just kind of keep an eye out for them. When they do start coming back, and be sure to water them and just help them they don't have roots like the flowering plants do, but they still need that support system, and that's how they get nutrients in and stuff. So yeah, I wouldn't be surprised.
It's so pretty.
Yeah.
Yeah.
Yeah, contractors are hard to deal with, even when you say, please don't go into that area. I've sectioned it off, and then you go back the next day, it's like, come on.
Yeah, I had to take off my birch grove.
Yeah.
Don't go here.
You have the conversation. I see that so many times. It's very
No,
frustrating.
they were great though.
Oh, good. Yeah. So, got another, yep. Yeah. With native plants, it's short. There are species of native ferns that have some remarkable fall color. Again, going back to hay scented fern, you usually get some really nice gold, kind of autumn gold colors. But in terms of having like that red color throughout the growing season, I mean that's kind of an anomaly and an aberration, and that's why it was selected to be a cultivar to grow out, because it's a pretty plant. I mean, I used to have some growing at my old house there, and now my dad has them, and they're still chugging along. Yeah. All right. Thank you, Don. Thank you for having me.